we have seen that energy transfer provides one quenching mechanism. If the acceptor is also fluorescent, then the transfer from donor to acceptor results in a change in the fluorescence. The original donor fluorescence is quenched, but the resonant acceptor then fluoresces instead. Fluorescence resonance energy transfer, FRET, is a distance-dependent dipolar interaction between the electronic excited states of two dye molecules, in which excitation is transferred from a donor molecule to an acceptor without emission of a photon in the transfer process. FRET is dependent on the inverse sixth power of the intermolecular separation, making it useful over distances comparable with the dimensions of biological macromolecules. Thus, FRET is an important technique for investigating a variety of biological phenomena. Let's examine the FRET between fluorescein donor and rhodamine acceptor. Note that I have offset the two spectra so that we can see how the donor emission overlaps with the acceptor absorption. When light is absorbed by the donor, it is promoted to its singlet excited state. Instead of fluorescing, the donor transfers its excitation energy to the acceptor. Then the acceptor emits a photon, but at a lower frequency. The acceptor emission is shifted to lower energy and can easily be distinguished from donor emission. There are three conditions that must be met to observe a FRET signal. First, the molecules must be in a distance range from 10 to 100 angstroms from one another. FRET is a long-range interaction because it involves coupling of two transition dipole moments. The distance dependence of a dipole-dipole interaction is 1 over r to the sixth power. R is the intermolecular distance here. Second, the emission of the donor must have spectral overlap with the absorption by the acceptor. FRET is a resonant process. The donor does not emit a photon that is then later absorbed by the acceptor. Instead, the donor transfers its excitation energy to the acceptor resonantly. Third is the caveat that the molecular orientation is important. Since FRET is a dipole-dipole coupling, the donor and acceptor dipoles must be aligned for maximum effect. If the donor and acceptor transition dipoles happen to be orthogonal to one another, the FRET signal vanishes. For the general case, the FRET signal depends on the square of the cosine of the angle between the dipole moments. The overlap of donor emission and acceptor absorbance is shown here. This example involves two fluorescent proteins, called cyanofluorescent protein, CFP, and yellow fluorescent protein, YFP. The FRET distance dependence can be expressed compactly as the ratio of R0 over R to the sixth power. R0 is the distance where 50% of the energy is transferred from donor to acceptor. It is known as the Forster distance. The definition of the Forster distance includes the quantum yield, the observed lifetime, an orientation factor, kappa, and the spectral overlap integral, J. Typical Forster distances for useful FRET pairs range from 20 to 80 angstroms. Fluorescent proteins provides many applications of fluorescence. This includes FRET because there are many differently colored fluorescent proteins derived from green fluorescent protein. Green fluorescent protein, or GFP, was discovered in a jellyfish. It receives light from a chemiluminescent process that emits blue light. The blue light is absorbed by GFP and then emitted as green light. This sounds like a curiosity, and it took years for the first applications to be realized. The GFP fluorophore is made entirely of amino acids. Here we see that it is a covalent interaction between glycine, serine, and tyrosine. Once researchers understood that this fluorophore was created spontaneously as the beta-barrel protein folded, they began to explore variations, using different amino acids and chemical modifications to make other colors of fluorescent protein. This is important because GFP can be inserted into essentially any gene. GFP will be transcribed and translated when that gene is activated. It is a measure of gene expression. Furthermore, GFP can be expressed as a fusion protein with a protein that localizes in the cell. When genetically encoded in this way, all proteins of a given type in the cell will be tagged with a green fluorescent label. One can see when the gene is expressed in the development of an organism and where the protein ends up in a cell. Here we see 4 other examples of modifications of GFP that fluoresce at various wavelengths. There are many such fluorescent proteins extending across the visible spectrum and beyond. One can also conduct FRET experiments with fluorescent protein pairs. This permits a scientist to observe when proteins associate in a cell. The FRET signal can be observed when 2 fluorescent proteins are less than 10 nanometers apart. If the proteins dissociate, the FRET signal vanishes. This type of labeling permits complex cell signaling to be observed in real time. Here we see the RAS protein releasing CRAF1. The FRET signal of a labeled protein will be observed only when CRAF1 is released into the cytosol and folds so that the FRET pair is close enough to support a FRET signal. Here we see the direct detection of protease activity in a cell using a FRET label.